introduce you Gerd Schröder-Turk. Gerd is in the Department of Mathematics and Statistics at Murdoch University, which is Perth, for those of you who know something about Australian geography. He's also an honorary professor at ANU, Australian National University, which is in Canberra, right in the middle. And then he spent 10 years in Erlangen, working on the physics of disordered systems, where he received an Emmy Noether Prize, presumably not for distinguished women in science.
Not quite.
So his expertise, as we're going to discover this evening, is in materials geometry. That's the use of geometrical tools to probe small-scale features that illuminate the properties of, for example, living systems. It's also known for drawing attention to the way that international students are treated by universities, something that's perhaps not restricted to Australia. So Gerd is visiting as part of this 4-week Isaac Newton Institute programme that is advertised behind me on geometry and packing, soon to come to a close. And the programme, of course, also marks 150 years of mathematics at Aberystwyth. Since the first head of department, Horatio Nelson Grimley, started teaching mathematics at what was then University College Wales, Aberystwyth. It's a slightly different name. So, it is with great pleasure that I invite Gerd to give us this lecture on saddle-shaped surfaces with biology and materials.
Hello, thank you very much, Simon, for that very kind introduction. And even like you've mentioned it, yes, I've spent the last few years doing things that weren't so much focused on physics, but kind of what I thought was just standing up for what university should do, provide good education. And it is very disappointing that it ended up being such a huge drama and so much pain. But I am very happy to say that at Murdoch things have greatly improved. And so I'm back at my normal space thinking about geometry and materials and that kind of stuff. There we go. So that's what I'm going to talk about today. And let's get straight into it. No, actually, sorry, let's not get straight into it. Before I start, I want to sort of just highlight that this is really a talk which sort of covers aspects from the last decade or so. And there are plenty of people who were involved. My former PhD supervisor, Stephen Hyde, lots of these ideas are his. Klaus Mecker, my boss back in Erlangen. Bodo Wilz, a very close collaborator. Matthias Saber, my photonic brain. Philip Schönhofer, who's done lots of surface assembly work. And then Annie Jessop, who's a current student of mine. And then I also want to take a moment to really thank the Newton Institute and Aberystwyth University and Simon and Adil and anyone else who was involved for being able to spend time here. It's been a fantastic workshop and I do absolutely love that library. I hope that library will be forever the center of this institute. It's just so gorgeous. All right, here we go. This is the man we should all admire, Alan Turing. He was probably, he only died a month ago or so. He was the one who invented the surface that I will mostly be speaking about, called the gyroid. And he was a real polymath. You know, he managed to do some very, very serious mathematics, even though, you know, by training he was a physicist. And then he ironically came to inventing this gyroid in parts by playing around with injection molding molds, because he was trying to make surfaces. All right, so here we go. This is the structure that I've spent most of my scientific life on. So this one is called the gyroid. It's what we call a triply periodic minimal surface. You can see it's got two sides, so it separates space into two parts, one the green side, one the orange side. Now you can see as you kind of fly through the orange side that there's lots of symmetry. There seems to be this helical element, and it also seems to have this branch structure, which is maybe reminiscent of a network or a labyrinth or a maze or something like that, right? So you can kind of turn left, right, up, down, and so on and so forth in there. Had we flown through the green side, apart from a mirror symmetry, it would have looked exactly the same, right? So this is why we call these structures bicontinuous. They divide space into two domains, each of which is a single connected component, and both of them are the same aside from this mirror symmetry that I described. Right? One way in which we will often sort of look at them is by basically replacing them by kind of the one-dimensional, we call them skeletal graphs, So these are like the networks that sort of trace the center lines of the tunnel network. And so you can think of alternatively these bicontinuous minimal surfaces as two intertwined lattices, in this case two intertwined lavas graphs, and then the surface in between them is kind of like the equipotential surface almost, if you want to think about them that way. All right, so this particular one was called the gyroid, as I said, invented by Alan Schoen 50 years ago, but it's part of a much bigger class of surfaces that are called triply periodic minimal surfaces or triply periodic bicontinuous structures, right? And there's a whole family of them, I mean, many families, so it's not just one structure, but there are three that have sort of become most famous in soft matter physics. On the one hand, the gyroid, and then two surfaces that were invented 100 years earlier by Amandus Schwarz. One which is called the diamond, because it's got the geometry of the diamond, and the other one is called the primitive surface, sometimes also called plumber's nightmare, because it would correspond to welding 6 pipes together. Yeah. All right. So the reason why I'm interested in these structures is because they occur in many biological and also many chemical systems. Right? And so I've here put up a number of biological examples where they occur, and it sort of starts up there with lipids that form in your tummy when you digest milk. Right? Then this one up there is kind of a lipid phase or a model for a lipid phase in the alveolar surface. So that's the surface between the air and the blood vessels in the lung. Then this one down here, that's a membrane which is found in a chloroplast precursor. So that's the things where photosynthesis takes place. This one here in the middle is an example of again another intracellular organelle, the mitochondrion in an amoeba. This one down here is a structure in a butterfly, and then finally this one up here is a calcite structure in a sea urchin. Yeah, and what's really quite remarkable about this is that you really are covering a huge range of length scales. Right? So these ones up here, where are we? This one up here, the periodicity is a few nanometers, right? So the channels would only be a few nanometers in size, right? And then it kind of goes up to the butterfly, where we will see the length scale is similar to the wavelength of light, and that's got some nice effects. And then finally, up here, I can't really see my cursor. Can I? No, I can. Sorry. Then finally, up here in this sea urchin, the structure is huge by molecular scales. It is in the 10 or 20 micrometers in size. Yeah. All right. So what I'm hoping to do today is kind of give you both a bit of an idea of where these occur in nature, jump into 3 of these examples in some more depth, right? And then also give you a geometric argument why they are good structures to form. Yeah. All right. So let's jump into the butterfly. So this one here is one of maybe half a dozen or a dozen species that form this structure. They are all of them green for some reason. And actually this green hair streak is quite common in England, and I'd love to find someone who can help me find the eggs and the caterpillars and the pupae. So if anyone knows the butterfly aficionado, let me know. All right, so if you look at it under a microscope, this is what the wing will look like, right? And you can see that there are these small, almost roof shingle-like particles, right? We call them scales, and they are the ones that sort of glitter, as you can see here, right? And if we want to understand them a little bit more, we need to go to electron microscopy rather than light microscopy. And again, you can see these particles, these scales. If you zoom in a bit further, you start to see some structure here, these longitudinal ribs that run along the scale. If you zoom in a bit further, you start to see some cross ribs that sort of connect neighboring ridges together. And if you keep going, you start to see that below all of that, there is a porous structure where, if you look at it for long enough, you can start to see some periodicities and some symmetries in it. Yeah? And it turns out that structure, and here maybe if you look down these channels, maybe you can even sort of start to guess that there are these helical elements. This structure is very closely related to this gyroid minimal surface that I showed you. Yeah, okay, good. So the way that this butterfly builds it is basically, well, that's not how it builds it, but that's how it looks, right? It's the gyroid minimal surface, then you leave one of the two channels hollow, and you fill the other one with a biopolymer called chitin. Yeah? All right. And length scale in physics is really a very important thing. There are a few things in physics that are length scale independent. And so this butterfly builds this gyroid roughly at the size where the periodicities kind of correspond to a few hundred nanometers, 300. And of course, to any physicist, 300 nanometers sort of sounds like something that's similar to optical wavelengths, right? And indeed, it turns out, if you think about what this structure may be doing in this butterfly, you may suspect, well, maybe that's the one that causes the color green through an interference effect, and you can do some calculations. This is what optical physicists do. So you come up, you basically implement a three-dimensional model of the structure. You model it made from chitin, which is very similar to most other polymers in terms of the optics. So it sees through, right? But otherwise, it's very similar, right? And then you can calculate. As a function of wavelength, right? You can calculate how much of the light that's falling onto the structure gets reflected, right? And you can see here, so this one is the reflectance. So you can hear kind of in the color that we actually see in the butterfly, so around kind of 500 nanometers green, that's where there's what you may refer to as a reflection peak. Yeah. So in this butterfly, the gyroid structure is responsible for creating the green reflection that you see, right? There's no, or there doesn't need to be any pigment in it at all, right? The green comes from the structure alone. Yeah?
If you had a different material than chitin, would you still get this color?
Yeah, the only problem is butterflies don't tend to use much else other than chitin. But sure, if you had a higher refractive index material, you would get a shift in the phenomenon in the wavelengths, right?
You would still get that.
You would still get the peak. Yeah. All right, I might show you a little movie that kind of demonstrates that, or at least gives some evidence, some indication that what I've said is true. So what you can see here is it running? Oh, yeah. What you can see here is one of those little dust particles, one of those scales plugged out, right? And then put under a microscope, and then we put some ethanol on top, right? And optically speaking, ethanol and chitin, they are much of a muchness, not quite, but close to it, right? So what has happened really is that the ethanol got rid of the structure basically by filling it all in with a material which is equivalent to the chitin. And so that's why you're getting very subdued reflections, very weak reflections, right? But then, of course, as the ethanol evaporates, which is what you're watching here, when the ethanol has gone, then the butterfly wing scale jumps back to its bright green state. Yeah, okay. So that's an indication that the color really comes from the structure rather than that it comes from a pigment, right? And of course, what happened there is you had a chitin structure, right? You basically get rid of that structure by filling it with ethanol, and I've chosen slightly different colors because it's not a perfect match, it's just a close match, right? So now the color is gone, or rather turned much lower in intensity and red, right? And then when the ethanol evaporates, the structure comes back and the color comes back too. Yeah, good. So that's what the butterfly uses this gyroid structure for. And of course, it's interesting because it's at a relatively large length scale. It is certainly at a length scale where we can think about copying the structure and using it, for some imagined photonic devices. this is one example of where we've done that. So this is work done with a couple of colleagues in Melbourne. They had basically a very cleverly designed stereolithography machine where they could basically print this gyroid at a length scale, which here corresponds to the telecommunication band, right? So we then put that into the shape of a prism. Good, and then we kind of checked whether we can use this gyroid shape to do something interesting to light. And in this case, this was because, as you've seen, this gyroid has this helical structure, so it's chiral. It actually turned out you can separate to an extent left circularly polarized light and right circularly polarized light. So you're kind of getting a prism that splits up light in that way. Yeah, but this was really just one of the applications. There have been, that's really a little subfield nowadays called gyroid photonics, where people have used gyroid structures to kind of think about what do they do to light and how can we use that to our benefit. Yeah. All right. So, of course, for those of you who have, I'm going to now come to kind of another system which forms the gyroid, and it's a system where I don't need to say too much because it's been covered by others in this workshop. It's the two areas of lipid and copolymer self-assembly. Right? Of course, copolymer self-assembly was covered in depth by Greg Grayson and Anne Shi, but I'm going to speak about it anyway briefly. Yeah. All right. So the question, I suppose, is why would lipids or copolymers or any other molecules want to form surfaces like they do, right? And in order to understand that, it's important to look at the type of molecules that form structures like this, right? And most of these structures are molecules such as a diblock copolymer here, where you've got a molecule which is made from two different parts, B and A. In this case, they are two polymeric chains, and these ones are connected to one another, right? And so this really means one half of the molecule is different to the other, and to varying degrees, they don't like each other. Yeah? If you imagined one of these was very much oil-like and one was water-like, then of course what they would love to do when you've got lots and lots of those polymers, they would love to separate with forming one big water phase and one big oil phase, but they can't do that because they're connected together. Yeah. So this macroscopic phase separation that I've sketched here, all the blues go to the top or the pinks to the bottom, that doesn't work. Yeah. And what they do instead then is what's called microphase separation, right? So trying to kind of build geometries where, you know, the blue chains mingle with the blue chains, the red chains mingle with the red chains, right? And in between them is an interface that separates them. Yeah, good. And as Greg discussed in quite a bit of detail, you know, you can get these hexagonal structures or cylindrical hexagonal structures, or you can have micelles and that kind of stuff. You can have lamellar phases, and as I said, you can, and as he said, you can have these bicontinuous phases, and in particular, the gyroid. Yeah, good. Similar mechanism, different system. So this is an example from a lipid or a depiction of a lipid water system. So lipids are molecules that we call amphiphilic. So their head group typically likes water better than their tail, right? So one is a fatty tail and the other one is a polar head group. And again, when you mix these molecules in water, right, then the tails would like to aggregate in a way that they are shielded from the water. Yeah? And there are multiple ways in which it can happen. It can happen by them forming micelles. It can happen by them forming planar membranes, which are separating water layers from one another. And it can happen by these molecules forming membranes, bilayers, which then get draped onto these three-dimensional shapes that I've discussed. Yeah. All right, and it's really quite exciting. So this one is a picture from about 6 or 7 years ago from a group in Switzerland, Raffaella Mezenga's group, I believe, and the microscopy techniques are now so good that you can actually look at these lipid phases as they form, or at least once they form, right? And you can see that under a microscope. And you need to remember here that length scale is really down to 10 nanometers or something like that. So this is really very, very beautiful. All right. Okay, good. So let's talk a bit more about why they would form periodic minimal surfaces. And let's first talk a little bit about some geometry about these minimal surfaces. So let's talk about them in terms of being subfilms. Yeah. So this one here is actually a screenshot from an absolutely wonderful series of videos that Alan Schoen recorded in the 1970s. You can find them on his website. One of them is preceded by his wife playing a piano piece for about 10 minutes. So it really oozes you into the into it. Right? And I believe these are his disciples, and he's trying to explain to them what forms you can get if you're dipping things into if you're dipping wireframes and so on and so forth into lipid into sub solutions. Yeah, good. And I'm sorry, this is a bit grainy. That's the quality of the video. But basically, the point is these minimal surfaces that I've shown you, you can think of them as being constructed from small patches that correspond just to normal soap bubbles, right? If you bent a piece of wire to be a certain suitable shape and you dipped it into sub solution, you would get a minimal surface, right, restricted to that wire, and you would, for example, here we've 3D printed one of them, and then you could take more and more of them and put them together, and ultimately you could form the minimal surfaces that are the minimal surfaces that I've shown you. Right? So here, two, they put them together, get a bigger piece, and you can take 6 of them. You can get an even bigger piece, and you can keep building any section of the gyroid minimal surface that you want from it. I'm sorry, this picture is so pathetic. Yeah. All right. I could. If time, I think we're good for time. I'll show you another little, because I realized I wouldn't want your daughter to get bored, Helen, so I'm going to show another video here. Let me see where that is. There we go. There we go. But you don't have to be interested. All right, okay. This is the video which was made by another colleague at the ANU, Stuart Ramsden, who also worked with Stephen Haidt at the time, right? And he's really just sort of reiterating what I just said before. So the gyroid is made from a surface patch, which is this triangular patch, right? You can take 2 of them and put them together, right? You can identify that there are some symmetries, in this case a point inversion symmetry, and that allows you to build this to replicate this piece in space, right? Then you've created an axis which corresponds to a fourfold inversion point, and that's also got a 2-fold rotation, as you can see here, and you could keep building this structure in this way from pieces, right? And as I said, each of these pieces you can think of as basically a piece of wire with the surface film spanned. Yeah. All right. So here we go. This is a kind of, I suppose, just a video where they're sort of flying and then they form a surface. At the time, we thought it would be great to commercialize this little puzzle. It's a puzzle toy, right? And so we had gotten some money to injection mold these pieces, right? And then we had this joining mechanism to put them together. And we were trying really hard to sell this, well, not to sell this, to get a puzzle company excited to help us commercialize. It was all in vain, despite these videos that they were created.
Given that piece, it's a unique way to assemble it.
Yeah.
And are there wrong ways where you can get stuck?
There are ways in which you can almost get stuck, but then they don't quite fit. Right? Yeah. No, it's a unique way. Yeah. Beautiful. Yeah. So there we go. So that's the gyroid minimal surface. And I suppose the next question that I want to, I mean, of course, we all know if we dip a piece of wireframe into soap, we're getting a surface which is area minimizing. Yeah. So as I said, this is built from small patches that are all area minimizing at the fixed boundary. Right? So of course, that immediately leads to the question or the assumption that probably the minimal surface as a whole is also area minimizing. And that's what I want to talk about next. So here we go. Right? So now this is not the gyroid, this is the P surface, but as I said, it's quite similar, and this certainly also holds for the gyroid as well. So this value up here, right? This is the surface area of the primitive minimal surface, as you can see it here, right? So what I'm now going to do is I'm going to sort of foliate that surface a tiny little bit, basically push it into one of the two channels, right? Of course, I can sort of push it a little bit more into one of the channels. So mathematicians call it a parallel surface transform, right? And you can keep going. And what you can see is that as you do that, as you foliate the surface, as you build these parallel surfaces to the original surface, your surface area goes down, right? So this is going into one of the two channels, right? Of course, I could try doing it going into the other channel, but remember what I told you is that the two channels are identical, right? So whatever happens to let's say the green channel will also happen to the brown channel, and you can see that here, if we foliate the structure but into the other channel, again the surface area goes down. Yeah, okay. So you certainly can't think of these bicontinuous periodic minimal surfaces as being maxima of the area functional under all possible deformations, right? Because there's this obvious deformation here, sort of pushing it, well, shrinking one channel and increasing the other one. And as you can see, the surface area goes down. Yeah. So with regards to that deformation, in fact, the minimal surface itself is a maxima of the area functional rather than a minima. Yeah? Of course, that doesn't contradict that it's a minimal surface. So it's got mean curvature equal to 0. If you drew any wireframe on the surface patch and dip that into soft solution, you would get an area minimizing surface. Right? So this is a matter of what deformations you allow. But what it shows is that there are some deformations, in particular this parallel surface transform, which makes the surface area smaller. Yeah? All right. Okay, good. So then, why would lipids or other simple molecules, why would they like to form these membranes? Yeah, because surely it cannot just be area minimization, right? So if you think about, water and lipids being mixed with one another, making a small-scale structure is never a good strategy for reducing the interface area, right? Because typically, when you reduce when you make the structure smaller, the interface area goes up. So it's not simple minimization of the surface area. So let's have a think what else it could be. Okay, and the answer is actually to a very large extent, we've already seen the answer on the screen, but the answer is also contained in a slice of toast or in a bimetal strip, right? So I want you to think of this one here as a bimetal strip, right, made from two different materials, a sandwich thing, a core which is sandwiched between two different green materials, right? And so then what I'd like to do is to cool the underside of this strip, right? So of course what this will do is it will contract the lower green strip, right? And just like a normal bimetal strip, it will consequently bend down. Yeah, good. So if you then say, all right, now I'm also going to cool the upper side, right? Then of course, it would like to bend upwards. The downwards bend and the upwards bend, they will cancel out, but in a stressed state, right? And the whole thing will become straight again. Yeah, I mean that's the idea behind a bimetal strip. Huh? So this is very much only true if you've got a one-dimensional strip, right? Because this final configuration that you've got here, right? While it is straight again, it is stressed, right? Because both the two green sides they want to be contracted because they're colder than the bulk, right? And they're only straight because one is pulling to one side, one is pulling to the other side. So when you do the same thing in 3D, you have additional degrees of freedom, and that's to form saddled surfaces, as you can see here, right? And that's where the slice of toast come in. And I give full credit to Stephen Hyde. He described that in 1998 in an incredibly beautiful paper called Boxing Patula. Right? Anyway, the idea here is very simple. And I'm sorry, the pictures are pathetic. I really need to.
I just want to say it's a very great journal as well that he was published in.
Well, that's right. It's actually he's just republished. He's republished it with additional commentary. So it's a great paper. Anyway, so what you can see on the left-hand side there is a slice of toast, right? It hasn't been grilled yet, right? And then if you start grilling it from the top and from the top only, right, then the top layer will dry out, presumably it will start to contract, and actually the whole slice of toast will start to curve upwards, and it will do that, in all directions. So it will actually try and form kind of a, spherical geometry. Yeah. Okay. But then if you flip it, right, and if you then also grill it from the other side, right, then suddenly both of the outer layers of slice of toast, they want to contract, but the bulk is still nice and fluffy and white, right? And instead of jumping back to the straight configuration, to the planar configuration, the slice of toast will actually jump into a saddle-shaped interface, right? And the reason why it does that is because the saddle-shaped interface, as we've just seen with the graph we discussed, is one which allows you for both interfaces to be contracted relative to the bulk. Yeah? So that's why you get saddle-shaped slices of toast. And just in case you want to try this, it is beneficial to cut the slice to buy thin slices and then cut it in half again horizontally. But it works. All right. So let's see if we can make this a tiny little bit more quantitative. Yeah. And let's think about if we can come up with the reason why minimal surfaces have got this property, always have this property that, if you go on one side of the surface or in the other, the surface area always shrinks, right? And in doing so, I think this is by far, from my perspective at least, the most useful way of thinking about curvatures. And I deliberately haven't defined what I mean by mean curvature and what I mean by Gauss curvature, because really that would come out of this little calculation. Yeah. Okay. So what I want to do here is I want to take a surface, in this case just a sphere, right? And then I want to inflate that. So I sort of basically want to do this parallel surface transform and translate every point of the surface by d along its normal, right? So in this case, of course, all you're doing is inflate the surface, right? And so if you want to know the area of the inflated surface, right? And of course we can calculate it. Let's call it A of d, right? So it's 4 pi times the radius of the inflated sphere, which is the original radius plus d square, right? And then you expand it. Turns out you can pull out a 4 pi r square, and what you end up with is a polynomial with a constant term, with a term that's linear in the thickness d, and a term that's quadratic in the thickness d. Yeah, and these two prefactors here, so the prefactor in front of the linear term, and the prefactor in front of the quadratic term, that's exactly the mean curvature and the Gauss curvature of the surface. Yeah? And that's a really beautiful way to think about curvature, because it basically means Gauss curvature and mean curvature are the two coefficients that tell you what happens to a surface when you move it when you move it off the surface, so to speak. Yeah? Good. So the beauty is that this also works. I mean, the example I showed you was a sphere, because the curvature is constant and blah, blah, blah. But it also works if you've got any shape, as long as it's reasonably behaved, you know. I'm not going to get into that. But so let's say, you know, for example, if we've got a surface like this dark blue one here, right, then you can pick an area element, you know, for example, this one up here, let's call that dA naught, right? You can basically extrude that area element, and you create a small parallel surface area element up here, right? And so for this area element of the translated surface, the same formula holds. So it's the area element times 1 plus now 2 times the mean curvature at the point times d plus the Gauss curvature at the point times d squared. Yeah. So you can do that in this local way. Of course, you can also do it in a global way. You can say, I still want to look at my inflated version of my entire object, right? So then what happens is that the area of the inflated surface, so the light blue line, becomes 1 plus 2 times the average mean curvature plus times d plus the average Gauss curvature times d squared. Yeah, good. And this works. It even works for concave. So it's not restricted to convex objects. It also works for concave objects up until you know what I've called here the pinch point. The mathematicians call it the reach, which is kind of the point up to where you can inflate an object and deflate it and recover the original object again. Yeah. So this is really a very beautiful way of thinking about curvatures, because it gives a completely intuitive notion to them, right? And for anyone who's a physicist, where of course you never have an infinitely thin surface, right? This will always play, or usually play, a role. Yeah. So if you're dealing with sheets of finer thickness, then that's what you get. All right, to come back to the slice of toast or the lipids or the bimetallic strip, here you go. Here's the picture that tells us why they do want to, or when they do want to form saddle-shaped surfaces, right? And again, I give full credit to Steve Hite, who kind of was the person who came up with this concept of the molecular shape for bicontinuous structures. But basically, the way to think about a lipid membrane is, you know, you've got your lipid membranes on one, your lipids on one side, the lipids on the other side, they stick together with their tails, right? Because that's energetically favorable for them, right? And then I suppose you will start to create these surfaces here and here, which Greg Bryson called intermaterial dividing surfaces. They're actually the surfaces between the water and the lipid membrane, right? So on these surfaces, then you've got the polar head groups which sit there, right? And then you can kind of imagine that you're basically depicting your molecules basically by their typical shape, right? So you're saying, all right, my molecules are defined A by kind of a length, right? They are defined by a typical size of the head group, and then they are also defined by kind of the width of the tails, right? But really, for the argument that I'm putting forward, the only thing that's important is that the tails in these molecules are wider, maybe by thermal fluctuations or because there's more than one, than the head groups. Yeah. Okay, good. And now it just becomes a packing problem, right? I mean, you can see that this is probably, you know, this configuration here with the gap here and the gap here and the gap there and there. That's probably not a very happy configuration for the molecules. And so rather they would like to adapt the shape of the interface so that these gaps disappear, right? So really we want to bend the interface both towards this side and towards that side. Well, we've been there before. That's exactly when you get saddle-shaped surfaces, because that's what they allow to do. Yeah, good. And we can make that more mathematical. So here's the formula that I showed you before, right? If we're requiring that the membrane is symmetric, so that going up is the same as going down, so A of minus D is equal to A of D, then that tells us that the mean curvature needs to be 0. That's why you need to get minimal surfaces. I'm going to gloss over the fact that this is averaged. And then if you want to have, in addition to that, if you want the polar head group, so this surface up here to be smaller than the mid-plane surface, then you want A of D to be less than the mid-plane, right? And that tells you immediately the Gauss curvature needs to be less than 0 in order for that to be compatible with that polynomial. Yeah, good. And so now we know why lipids form these negatively curved bilayers, and the answer is simple, because lipids with a narrower head group than their tail, when they form membranes, they prefer negative curvature because the interface then fits their shape better. Yeah? And so that's really the whole story behind the negative curvature, negatively curved lipid membranes. Yeah, good. All right, here we go. Good. All right. Let me dig a little bit. I'm not really, I know I teach mathematics, I'm even called a professor of mathematics, but I'm sort of a physicist really who is on the computational side and on the theoretical side. I just happen to be teaching mathematics. So I'm not really a hardcore mathematician, but let me nevertheless dig a little bit into minimal surfaces in relation to hyperbolic geometry, and in particular in regards to Hilbert's theorem, which we've heard once or twice during the last few weeks. Okay, good. So, and again, I'm doing that because we'll see it has some bearings on why they form in these lipid membranes. Yeah, okay, good. So this is some further very beautiful work, which again originated from Stephen's kind of school and was largely carried out by Miff Evans, who is nowadays a professor in Potsdam, but there were several others involved: Stuart Ramsden, Vanessa Robbins, Martin Peterson, and others. Yeah? And so what this shows here is a way of taking the hyperbolic plane and tessellating it with some tiling, right? And then basically mapping that tiling of the hyperbolic plane onto the gyroid minimal surface. Yeah, okay, so you can kind of come up with a one-to-one map between the two. And that sort of gives you the idea, which certainly is, that's Steve's religion, that you should, think about these negatively curved structures basically as images of the hyperbolic plane. Yeah, good. So what is the hyperbolic plane? At least to the extent that I want to think about it, it's really basically it's a 2D plane, right? With a metric which is chosen such that every point has constant negative Gauss curvature. Yeah? So the way that this is then portrayed here in this Poincaré model, of course, is that really in the true hyperbolic plane, all of these tiles are the same, right? But because of the way that we are representing it in this Poincaré model, they appear to be compressed here at infinity, and they appear to be smaller. But really, the metric is such that everywhere you've got constant negative Gauss curvature. Yeah. Good. And then there's Hilbert's theorem, which Keim also referred to, which basically says that there's no complete regular surface of constant negative Gauss curvature in R3. Yeah. Okay. So if you're hoping to find a way of embedding this hyperbolic plane in a surface in R3, it cannot work. Yeah. Good. Except, of course, that we have seen, Keim showed these images, this beautiful construction which he had from these pieces of foam, right? And which, as far as I understand, really they mirror this very lovely work of people who have crocheted hyperbolic surfaces, right? And if I'm not mistaken, this was pioneered by Daina Taimina, who I think was or is a professor at Cornell. Yeah. Okay. So by the construction, as Keim explained, these structures do have negative Gauss curvature, right? So So, you know, this sort of seems to be, at first glance, a problem with the Hilbert theorem, but of course it's not. And the way that both of these models work is basically by kind of finding a mechanism where you recognize the the perimeter of a circle in the hyperbolic plane is always greater than 2 pi its radius, right? So the perimeter grows faster than the radius, right? And so, you know, you add additional stitches in the crochet, right? Or here you have this mismatch between the between one side of these foam strips and the others, right? So that's how you get negative curvature here, and because you're always doing the same thing at every point, you're getting these patches, right? All right, and of course, Keim has, you know, this is from his website. He has put this up as what is up with the Hilbert embedding theorem anyway. And so the answer here, of course, is that yes, the construction is such that you get constant Gauss curvature, but eventually these structures will self-intersect, right? So you don't get an embedded surface as you would like it. Yeah. All right. And so that, in some loose way, it means you can think about these two differences that I've talked about now in two ways, right? So you can have the desire to embed H2 in R3, right? And you can, you know, there's no free lunch. You won't be able to do it in the perfect way, right? So you can do it in two ways. So either you can do it as shown up here by having a surface which locally has constant mean curvature, but it won't be an infinite surface. It'll have self-intersections, right? Or you can kind of do it in the way that I've, you know, that Miff and Stephen and others like to think about it. You can embed it on a negatively curved minimal surface, right? And then you know, well, it's embedded, it's infinite, it's beautiful, but you know that there must be variations in the Gauss curvature, right? Because you can't have a surface which has no variations in Gauss curvature. Right? All right. So of course, when we're thinking about membranes, lipid membranes, then they don't tend to self-intersect. They also don't like to have boundaries. So really, this one down here is probably the more realistic way for thinking about that. Huh? And that brings me to the question then. So we've done this average curvature analysis to understand why lipids may want to form negative curvature. But I want to just spend a couple of minutes talking about why do lipids want to form the P, the D, or the G surface, right? Can we understand what sets them apart from the many other surfaces that are also minimal surfaces, negatively curved and embedded? Yeah? Good. And that comes very much to the title of the talk, but we'll get there in a second, right? So the argument here is really very simple, right? So if you are intending to build a membrane from lipid molecules that are all the same, right, then chances are that they will want to form an interface which is the same everywhere, right? If the lipids are the same, then presumably that means they would like their local environments that they adopt to be the same. Yeah. Okay. Good. So this, in a very qualitative way, then motivates why we start to look for why we try to address the question, which of the surfaces that have negative curvature, so they fit the average shape, which of all those surfaces is the one that gives that has the greatest uniformity in curvature, right? Because presumably that's the one that lipids and other molecules would then prefer. Yeah. All right. And unfortunately, we haven't got, you know, I can't say the gyroid is the winner because, you know, we don't have an ability to test all possible surfaces. So we kind of have to, you know, kind of start from a set of test surfaces and analyze them, right? And this is what this graph here shows. So this is the, you know, I suppose the landscape where you've got the cubic gyroid here. You've got the primitive surface here, you've got the diamond surface here, and then as lines, you've got a whole lot of surface families indicated. These are surface families that people have parameterized, right? And for particular values of the free parameter, they happen to be the G or the P or the D. Right? And so this seems like a natural sort of set of surface families to kind of start our analysis with and say, well, amongst these families that contain P, D, and G, are P, D, and G optimal? Yeah? Good. And just to kind of give you an idea what these surface families are, so one of them, this one here, the TD, is, the TP, this one here, is one where you start with the P surface, right? And then either you stretch it in one direction or you compress it in the same direction, right? You can do that, and if you let the surface relax, then it will adopt minimal surface configurations, such as the one shown here. So this is the undeformed one. This is one where you've compressed it. This is one where you've extended it to some extent. Yeah? And the other surface families that are shown here, they are kind of a similar type, but mind you, they are a bit more interesting in terms of what they do to the surfaces. All right, okay, good. And what we do then, very much in a physicist's way, is we'll say, okay, let's just define the like a variance. Sorry, where am I? Where are we? Yeah, right. So let's just define the variance of Gauss curvature over the surface, right? And let's study that as a function of the free parameters for each of those surfaces. Yeah, okay, good. And so what you can see is that these curves, they do tend to have minima, right? And the minima tend to be at the cubic surfaces. So here at the G, here in this surface family at the P and at the D, here in this family at the cubic D, and at this family at the cubic G, and this last
using a different mouse.
Oh, am I? Oh, sorry. Huh, funny. All right, that's what. Yeah. So, all right. So here we go. This is the cubic G in the RG family. This is the P in the RP family, and the cubic D in the RPD family, and so on and so forth. Right? And so, without going into any of the details here, amongst these relatively simple surface families, the cubic P, D, and G are the structures that have the smallest degree of Gauss curvature variation. Yeah? Good. We did also carry out another analysis which relates to what Greg Grayson was talking about, because you can also ask the question, is it just variations in curvatures, or is it perhaps variations in the thickness of the channels, right? How much do the channels vary from one spot to another? And that's what's done here. So this is the variations of the channel throughout the structure. And when you do that analysis, then it turns out that the gyroid, right, here emerges as the winner. So the gyroid is the one that alongside P and D has the lowest degree of curvature fluctuations. But amongst our three surfaces, it's the one that has the most uniform channel thickness. Yeah, good. All right. So I wish I could announce a more definite answer to, is the gyroid nature's best attempt at embedding the hyperbolic plane in R3? I can't, but I can say amongst the limited set that we analyzed, P, D, and G come out as the best in terms of curvature distribution, and that's no surprise that they are the same, because they are, in some sense, they can be mapped onto one another, right? And the G does win out of all of them when it comes to the variations in the domain size. Yeah. So this one here, I think, is a question which it would be very lovely if there was a, some further study of it, or perhaps even some mathematician to sort of pick that up. All right, so let me finish off by just covering a couple more examples of where these structures occur, right? And I'm going to show this to you because these two examples are now at a different length scale, right? Remember this argument which I gave you for packing, that worked when the molecules were a few nanometers in size, right? So, of course, now if you imagine the same molecules forming a structure which is 100 or 1,000 times bigger, suddenly the energetics become different, right? So this simple packing argument probably worked well while the sheets were relatively thick compared to the volumes. right? Okay, so here's one in plant membranes, and I'm really only going to flesh it up. So this is a runner bean. When you look at the cell that would become the chloroplast, so the one that has the chlorophyll in it, if you look at it while you grow this plant in the dark, or while you grow the seedling in the dark, it will form a diamond structure. If you then turn on the light, right, that will jump to the much more conventional grana stack structure where the chlorophyll sits. Right? No one really understands why it happens or what the plants use it for, but it does happen. Right? And it happens again at a length scale where the length scale is now so large that these very simple packing arguments probably don't fully explain anymore why these ordered membranes occur. All right. And the other example I wanted to briefly flash up is the one of the sea urchin. So this is some tomography that we've carried out on a sea urchin from the North Atlantic, I think. Right. So this one is, I suppose, kind of just the tomography data set of the whole of the sea urchin as a whole. Right. It's got all that beautiful structure to it, of course. And what we'll do in a second is we'll kind of focus on one piece of the plate, which is also called stereom, and then we'll look into that a bit more. Yeah. So here we go. And if you now look at it kind of from the side and cross section, right, then you start to see that there is this rather kind of porous structure here, and again, it seems to have some regularity. Right? All right. Okay. This is also something which people have studied for a long time, so it's no surprise that there are these structures, but I suppose the question of why these structures can be so ordered is one that's still open. Right? And so what we've done here is we've actually looked at it, and we've kind of picked up one little piece here, and we extracted the tomography data from it and fitted it to a surface model, and with quite high certainty we can say that is, as had been reported before, the primitive surface. Right? But then when we went to a slightly different piece further down in here, and we extracted the piece of data, it turns out that piece can be fitted with even greater certainty to the diamond minimal surface. Yeah? And if you go another bit further down here, you find a structure where there's no order or periodicity at all, but it's got the same bicontinuous nature. Right? So why this sea urchin forms a composite material where it would use different types of bicontinuous geometry throughout its shell is unknown, and it's really fascinating. And it is happening at a scale of microns, right? 10 microns. So this is more than 1,000 times bigger than the curvature-based explanation that I've given you for the lipid membrane. All right. Okay. Good. So I'll leave it here. I hope I've shown you a few interesting examples where these minimal surfaces occur. I hope I've convinced you that you can, when you correlate interface curvatures with the shape of the molecules that make up the interfaces, you can come up with a picture why negative curvature is favored in some molecules, and also why homogeneous or uniform curvature distributions are good. As I said, the formation principles at the larger length scales are unclear, and it'd be really fascinating to, even from, I suppose, a purely mathematical point of view, to think about it. Right? And it's probably far-fetched, but similar structures have also been found in some numerical models of reaction diffusion systems. Right? So it's possible that there could be other explanations. Right? And of course, when you're talking about biology, there's always the possibility that it's not just an accidental byproduct, but that it's actually selected for fitness or purpose. Right? And so you could ask the question: is there an evolutionary benefit for the sea urchin for having this diamond structure? Right? It'd be pretty subtle, but it's quite possible. All right. So with that, Thank you very much for your attention.
I've always been fascinated by that network-like structure in the plants, which goes from being in the dark and then being able to synthesize machinery. Can you say a bit more about that? What the current state of understanding is? You said there was something not understood yet. I just want to hear a bit more about it.
Yeah. So the really the person to ask is Lucia Kowalewska. She's running a lab in Warsaw. Who's studying this, and she's very much studying this from the perspective of why is there this structural transition, right? So what she finds is that it occurs and it is reproducible and it happens in lots of plants, right? It is also to a degree reversible, right? So when you grow a seedling first in the dark, right? And then you expose it to light, right? And then you put it back in the dark, the grana flakes disassemble and reform the bicontinuous structure, right? So that bit is I think it's established that it's a real phenomenon and that it's reproducible and that it happens. The biological reason for it is really unclear, right? Of course, you could think about, day and night is obviously something where it periodically goes from light to darkness, but I think that, it's not totally obvious, right? So it's not like you.
Does it pack up all the way to that point on the left in the course of a few hours, and then when it comes day, it has to unpack all of that stuff on that?
Yeah. As I said, just to think it's a day-night cycle, that's too simplistic, unfortunately. Sounded great, but it's too simplistic, I believe. So yeah, no, it's I don't know. Okay. But I do believe this is the system because really we don't understand why biology uses these membranes, right? And I believe this is the system which is both experimentally accessible and interesting enough that it can be kind of a Drosophila fly, I suppose, for understanding why you get these bicontinuous membranes.
Do we make them as humans and use them in industrial applications like chemical processing or something? Kind of look like human.
We do to an extent. So I suppose, well, we're certainly using these mixtures of lipid and water for various formulations. Right? And there are some applications where people have, there's a company in Sweden that's creating small particles with these lipid nanostructured phases inside for the purpose of drug delivery. Right? And so the idea is kind of you load them with drugs and then upon temperature or pH change or something in the body, right? It releases. So there's plenty of that, or not plenty, but there's some of that. They certainly have an interesting rheology, right? Because the membrane forms a connected domain, right? It's actually more gel-like than if the lipids just form micelles, right? So there's plenty of it. But I do think that probably what I'd love to see happen in the next decade or so is a better understanding of why and where they occur in biology. Right? So there's a group in Copenhagen, Ben Boyd, and they have been looking at the formation of these phases as you digest milk. Right? And we know, or they know, that phases like this form. We don't understand yet exactly why, if there's a benefit. So I think that's the kind of thing where I think hopefully there'll be some progress.
I believe that you mentioned a few times about it's no doubt why the nature of biology goes through this. I mean, there's not
hot topic in engineering, it's also applied mass, it's metamaterials. So we have the lead, we have some small elements, and then we have purpose. So then you ask this small element to create a structure to make this purpose better or worse. And then as a result, people constructed some structures. And this structure actually time to time following some rules. So I believe that it's not that it will be 10 years. I think it's something that is very close to community. So simply asking this particular plant why this plant, so what this plant minimizes to survive in the darkness or in the light, so probably will be the answer for the direct structure. So it's purpose.
Yeah.
I was going to ask the question, are there animals that use this sort of ethanol-like trick to hide themselves when they're hunting and drain the ethanol out when they're displaying?
Like butterfly breathalyzer tips. Yeah, maybe. I don't know. There are some groups that are trying to create, trying to use these photonic effects in porous photonic crystals for the purpose of gas detection. So, because in principle you can, change the color ever so slightly if you change the gas that fills the channels. Right? So there are some attempts to do that. Yeah.
Curious about why your idea of commercializing with people work. Was it too complex? Or is there any particular reason, or it's just?
Well, I mean, there was a toy which we were hoping to develop, and it was, our joining mechanism for the parts was a little bit clunky.
Technical stuff.
With the stuff that Greg Bryson showed the other day with magnets, we later tried that too, and they gave a much better mix, but we didn't have the money to do another injection molding one. So it was fun, but
3D printing?
Yes, but ideally you'd like to have a few hundred pieces for every part in the world. That's it.
Question regarding structural color.
Yeah.
You talk about it being created by processes that are reasonably ubiquitous in nature. And you've given some examples, things like butterflies. I think earlier people talked about bird feathers as being some very strong examples of structural color. But if you go for a walk in nature in the woods, or how ubiquitous do you think structural colour is as a phenomenon? What contribution, if I pick an apple, could have been on there? How much structural colour is there in apple?
As opposed to pigment theory?
As opposed to, well, I come from the background as a psychologist, and we used to think about specular surfaces and Lambertian surfaces. And there was really nothing else. And when I came towards the end of my time as a psychologist, we were becoming aware that actually what's true for how we see a piece of paper might not be true for how we see more complex objects, such as a piece of fruit or a piece of wood that's in what a simple uniform surface. What we want to do about
So I suppose the one thing you can do with structured color, which you can't do with pigment-free color, is things like iridescence. Right? So that the object looks different depending on the angle at which you look at it from. So I suppose probably most, if not all, iridescent biological materials, they would be in some form structured. Mind you, in most of the cases, probably much simpler than here, multi-layers of some sort. It's very interesting. So in these butterflies, right, it tends to be the green that is, well, all the gyroid examples that we know are green, right? They're very few, if any, examples where red or yellow or brown is structured in the nature. So, yeah. it's, yeah, as well, whether or not the insects can tell a difference between that type of color, well, that's hard to say. Of course, if you record a spectrum, you're going to see a spectrum that corresponds to the structure. It's the specific spectrum that corresponds to the gyroid structure. Right? Mind you, this butterfly that I showed, it's believed that it uses the color green for camouflage. Right? So it's kind of not that dissimilar, really, to the typical green of the environment where it lives. Yeah. There are some examples of structured color, and I think we saw them yesterday in Gareth's talk. These beetles that have extremely strongly circularly polarized signals, right? But any attempt to find any biological purpose or even receptor in the predators or in the beetles themselves has failed, right? So that's, sometimes structured color is also just a byproduct of some other function.
What happens if you cover up a little spot on the beetle with the circular polarized light and left it in the wild and see what happens to them a year later?
Yeah, I mean, well,
after 3 months in there.
I mean, people have done on those ones because the effect is so striking. People have done behavioral studies, and they have found nothing.
Okay. Has anybody ever looked at colour constancy in these surfaces?
Color constancy.
Colour constancy, usually, for example, if you take a yellow light or a blue light and you shine it on a piece of paper, what reaches our eye is the combination of the reflectance and the colour of the light source, or different properties of the light source. Where exactly what the same drop or colour is breaks that law, because the light that reaches your eye isn't a product of the colour of the light source multiplied by the reflectance of the surface, but it's actually independent of the light source to a greater extent.
Good question. I don't know. I'll ask Bodo. Yeah.
I think the butler has put out the wine. Good time, but we were welcome to join for a drink, and thank again for wonderful lecture.